change in time zones. You know that in US it is near about midnight now. So he has graciously accepted our invitation to join and deliver his talk. So I humbly request him to start the session for today. Dr. Syed.
Thank you. Thank you for your introduction. Let me see whether I can share my slides. Um, Everybody can see my slide?
It's visible, sir. It's clear.
Okay, that's good. So, thank you very much and for the kind introduction. I think there may be some feedback loop. If there is any problem with my audio, just somebody says stop, and then I will stop it because I don't see any other screen other than my slides. So, for today's talk, I'm going to talk about nanomechanics of biomaterials. And my background is I'm a material scientist and also well experienced in developing instrumentation for characterization of materials. So I'm not a biologist. But throughout my career, when we were at Hydrotron later at Bruker, we interacted with many biologists to develop instruments, and we collaborated with them. And what I'm going to present today is part of those kind of collaborations and our own work that we did at Hydrotron. Okay, so the way I am dividing my talk today is in two parts. The first part, I'm going to deal with testing materials, hard materials, generally materials like bone, teeth, and things like that. And later, I will switch over to soft materials, and that's the area that I am presently focused on. The reason why I have to divide like this is I'm going to present a technology that we call depth sensing nanoindentation. That technology was developed originally for hard materials. So testing biomaterials, bio-hard materials wasn't a big problem. With existing equipments that we had, which we built about 20 years ago or 24 years ago, we could seamlessly just go ahead and use it, and we can get some reliable numbers, and we can characterize accurately. But over the years, we realized that the instruments that we developed for testing hard materials may not be sufficient when you come to measure properties of soft materials because of its unique challenges. So we kind of reinvented, redeveloped, and even now it's kind of very hard area where we are continuously developing models and instrumentation to tackle some of the problems that we'll face with testing soft materials. So that's why I divided it into hard and soft. So one extreme, it's hard bone-like materials, and another extreme, soft like brain, tissues and cells and things like that. And with respect to instrumentation, there are two types of instruments normally people use. If you are length scale that you wanted to measure is micro and nano. One type of instrument is atomic force microscope, and the other type is Nanoindenter. Today, I'm going to talk about only nanomechanical testing using Nanoindenter. Atomic force microscope is being well used and well recognized in biological testing of biological materials. But I'm not going to cover that. Maybe even at the end of my talk, I will present a few things which are unique about atomic force microscope. But to start with, both instrumentation, what we are trying to achieve is try to apply forces and measure displacements. So, and from the force and displacement measurement, we try to come out with how to characterize materials. The advantage with the atomic force microscope is that because the sensitivity is so high, you can apply very small forces in the orders of piconewtons, and you can study cellular level, like DNA level, you can study without any problem. But when it comes to higher scale, length scale, like cells and tissues and organs, we would like to use Nanoindenter as an instrument to measure those properties, mainly because of its quantitativeness and the range of force that it can apply and the displacement that it can measure. So that's how I'm dividing my talk. So I just mentioned that nanoindentation, but I think before I jump into it, I just wanted to kind of give you a background. Whenever we try to measure, today I'm going to talk only about mechanical properties and mechanics. So when we talk about mechanical properties, normally all the engineers we know that you're used to and you're trying to do tests like tensile test or compression test, and you're very familiar with that. And properties like yield stress and Young's modulus should be very familiar to you. But those kind of tests, although we can down nowadays we can do down to nanometer length scale, they are very difficult. Okay, mostly it is used for testing bulk materials. But if you wanted to understand properties at very local and very site specific, you cannot use those tensile or traditional compression test. So in such cases, depth sensing indentation technique, which I originally was developed for understanding the properties of metals, mainly thin films, coatings, and things like that, can be directly used to understand properties of site-specific properties of materials. Okay, so and there is a clear distinction between these two techniques. Both of them where you are applying forces and measuring displacement, whereas in indentation technique you are applying force through a sharp probe and measuring the displacement of the probe into the sample. Okay, resulting is basically you get a low displacement curve, and from that you try to get the mechanical properties. That is what you're trying to do. Okay, so going to the next slide, I'm going to show you a small video that kind of pretty much explains what nano indentation means. I'm going to show this video. You can see a sharp probe kind of indenting on a sample, loading and unloading. You can create a dent here, and then on the side you can see this low displacement curve, and that gives you the idea of the force that you are applying and the penetration depth that you can apply. Okay, so this is essentially what we do when we try to understand the mechanical properties of hard materials. So the probes are pyramidal in shape, and they are very sharp, down to like 30 to 40 nanometers in diameter, and they are made of diamond. It's very hard material. Okay, to apply the force, you need a transducer, and there are various ways of doing it. The best way is by doing electrostatic force actuation. That is when you have like this. I'm showing a transducer over here. You can see there are three plates. The center plate is suspended by a spring, and there are two outer electrodes, and there is a gap between these plates, right? So now if you apply a high voltage at the outer electrode, bottom electrode, it is going to generate electrostatic force, and then it will attract the central electrode downwards. And the indenter probe is mounted on the central electrode, and that's how you create force, and that force is being applied onto the sample, right? Now, this, if you know the physics of it, you can see that there are like parallel plate capacitors, and the center plate can move in between these two outer plates, which means that the capacitance can change. And if you have a way of measuring the change in capacitance, you can measure the displacement of the central electrode. So this transducer can be used to apply force as well as measure displacement. So it's a very compact, simple, very simple actuator and a sensor. Okay. So down here, I've shown the load displacement curve, and if this is the loading part. As you load, go to the maximum force. When you unload, you can see like a straight line, but it is not straight line. And you can measure the slope of this load. So when you measure the slope, it's basically load divided by depth. It gives you the stiffness. Okay, and stiffness is what you need to deconvolute the mechanical properties from these kind of curves. Okay, so I'm going to probably show in the next slide how I just mentioned how the force is generated, okay, and how it is applied onto the sample. And by electrostatic force means you apply a voltage, high voltage between the central electrode and the bottom electrode, and the force is basically voltage squared times a constant. So this is a very simple equation, so you can calculate the force if you know what the voltage you applied. So the force determination is pretty easy. I also mentioned that we can measure the change in capacitance and then convert into displacement. This is the equation for that. Because there are two plates, outer electrodes, and there's a central electrode, you can form two capacitors like C1, C2. So if you take the difference and divide by the total capacitance multiplied by the voltage that you apply, it directly converts into displacement through a calibration. The force and displacement can be measured this way, and we can get the load displacement curve that I presented just a little bit ago. Now, this is a unique transducer, and it's a very small transducer, so that it can mount onto a piezo actuator, normally a 3-axis piezo positioner. Those who are familiar with atomic force microscope, they can really recognize what is a 3 piezo actuator, the tube actuator. And when you apply voltage in X and Y direction, it can move in X and Y direction. And if you apply the Z voltage, you can move it in Z direction. So it has the capability to move in X, Y, and Z direction. And it can go down to nanometer length scale. So it can precisely move to nanometer length scale. Now, if you mount this transducer that I just mentioned on top of this piezo positioner, you can use it as a scanner. For example, when you scan with X and Y, by applying voltage in X and Y direction of the piezo tube, and by making contact with this transducer onto a surface, as the transducer slides in X and Y direction, it is going to feel the surface roughness, ups and downs and bumps, and then the transducer will move up and down, that you can measure as a voltage. Now, if you do this line by line, you can create a 3D topography of your surface. That is the advantage of this system. Now, you can do this kind of topography down to like few nanometers. And so you have a nanometer resolution, high resolution scanning probe microscope built into with a Nanoindenter system. So we call it scanning Nanoindenter. So not only you can use it to apply force and measure displacement like doing inundation, you can also use it to scan on a surface and get an image of the surface. Here in this slide, I'm showing on the right side that you can see the images. The first image is of a wood cell. You can see the cellulose structures, and you can clearly see the cell wall. And you can see in the rounded, like in red markers that I have put, you can see small dots. Those dots are basically indents that's been placed all across the, all along the cell wall boundary. So this is kind of a typical example I'm going to show you, where you can use this technology not only to see the surface by scanning probe microscopy, but then you can also pinpointly locate where you want to do the test and measure the force displacement response. Okay, so it is kind of a microscopy, scanning probe microscopy you have in situ with your nanomechanical Nanoindenter test instrument. So you have both the capability within one single instrument. Okay? Now this is another image where you can see a morphology of the surface, which is this is a two-phase ceramic material, and you can see the ceramic part as well as the matrix, which is a epoxy part. And you can also see there are triangular indents being placed, and these are the locations that you can choose to do the indents. So this is the SPM image that normally you get. And with that, I also mentioned that you can do load displacement curve, measure the penetration depth, measure the stiffness of the unloading curve. Once you know the stiffness of the unloading curve, you can measure the modulus of the material because by calibrating the tip, you can get this the area of the tip as a function of depth. We call it as area function. And once you know the stiffness in the area of the indent that you did, you can calculate the modulus through this equation. And if you take the maximum load divided by the area of the indent, you measure a parameter called hardness, which is nothing but mean contact stress. So you can measure two properties. One is the plastic property of the material, the plastic deformation property of the response of the material, which is hardness, and the elastic deformation of the material, which is the modulus. These are all the two fundamental properties that you need to know to access the mechanical strength of material. Okay, this is something similar to when you do a tensile test. You know what the Young's modulus is, but yield stress is something similar to what you measure with hardness. In fact, 3 times yield stress is hardness of the material. So you can correlate to your old yield stress and Young's modulus using this inundation testing scheme. Okay. Now, this is all well and good. Now, again, this particular curve that I'm showing on the right shows 3 different curves. We call it as mechanical fingerprint because each load displacement curve that you get is unique because it shows the mechanical response on that particular site or the material. And you can use that as a fingerprint to identify material and characterize. Okay. So this is all good, and most of this technology was developed to understand ceramics in metals. Like I mentioned, on hard materials, it's not much of a big extension to use this technology to understand the properties of biomaterials. So let's go with this slide, where I just briefly mentioned about the whole the AFM, what AFM can do, and also mentioned about Nanoindenter, what Nanoindenter can do. But all these techniques, it depends upon the length scale that you wanted to work with, right? So for example, the micro indentation, people may be familiar with optical based micro indenter, where you do the same experiments but use an optical microscope to see the indent size and measure the hardness. It can go up to certain length scale, but beyond that you cannot go because you cannot see those indents in optical because optics doesn't have the resolution. Then you have this Nanoindenter, then you can extend this to almost to hundreds of nanometers or less than 100 nanometer in dimensions. Okay, but if you want to go beyond sub nanometers or to the single nanometer, you need to have something like AFM. So to cover the length scale, you need to have a certain type of instrument. Okay, with a Nanoindenter, actually we can go almost closer to AFM, like 10 nanometer or less than 10 nanometer. We can do, and we can go up to 100 microns, hundreds of microns in depth. We can do that with Nanoindenter. Okay, so choosing an instrument also depend upon the length scale that you wanted to make measurement. Okay, normally with Nanoindenters, you can pretty much cover most of the length scale that that is of interest with respect to biomaterials. But when it comes to like DNA, if you want to sense the DNA level forces to split the DNA and things like that, you probably end up having atomic force microscope for that kind of application. Again, I can show I'm showing here in this picture. There is a length scale with respect to in biomaterials as well. For example, when you talk about organs, it's in centimeters length scale, right? And tissues are in millimeter length scale. And cells and things like that are about in micron length scale, okay? So depending upon what you wanted to study, the type of instrument also important, like I just mentioned, but also the probe size also important. For example, in Nanoindenter, you can have a micron size probe. At the same time, you can also have nanometer length, like 30, 40 nanometer tip radius probe. So choosing the probe also depending upon which length scale you wanted to study. Do you want to study at cellular level or tissue level or much higher level? Okay, so that is what is important. So you have to correctly choose what you want to what type of instrument that you want to use, and what is the shape and what is the size of the probe that you wanted to use, depending upon what you have as material. Okay, so like I mentioned, let's start with bone. And for the bone, we will use three-sided pyramid indenter, Berkovich, which is commonly used for understanding properties of metals. Again, this probe is made of diamond. And typically, you get this kind of low displacement curve. And I mentioned that you can measure hardness and modulus from that. Okay. Normally, the way I am classifying this as hard and soft is based on the elastic modulus. For example, when you have modulus about 1 GPa, it's like typically for the modulus of bone. And then if you go down, and like when you have cartilage type material, it's about 100 MPa, and below that is in kilopascal, zero 0.1 MPa. Okay, basically like contact lenses or brain or the cornea or veins and valves, heart valves, they are like 100 kilopascals around that range. Okay, so those materials I call it soft, and other and just a minute, sorry. Okay, yeah, so I call from up to like 100 MPa, I'll call it as hard, and below that I call it as soft. Okay. Now I talked about bone. I think we'll start with bone. Bone has a unique structure. Okay, it's a hierarchical structure. I have given a schematic here on the left side. You can see that, and you can see this ring-like osteons, and it has a lamellar structure actually. Okay, so the properties of bone is very important to understand, because with aging, bone becomes weaker and weaker, and also there are bone diseases, basically osteoporosis or arthritis, where the bone becomes weak, and it is subject to like fracture, even at small forces. So understanding the mechanical property of bone is very important. But if you look at the microstructure of the bone, it's very complicated, right? So if you take a big chunk of bone and do a macro test, or how that we have been doing in many years ago, you will get a number, but it doesn't give you a full understanding of the underlying microstructure and what it influences, right? So because now we have technology where we can go down to nanometer length scale, we can go down to this microstructure, understand those properties, right? So for example, these osteons, and they have a kind of a tubular structure, and so lamellas, basically it's a layer on a layer like onions, right? And depending upon the orientation, how you do the test, the properties may be different. That's why I call this slide as anisotropy of bone lamella, right? So it is important to understand, like when you have a load in a particular direction, what is its influence in terms of its elasticity? When you measure modulus, modulus is basically a measure of elasticity. How much load that structure can take elastically without fracturing or going through any deformation. That is the motivation that we want to know. So now with this instrumentation, we can go and understand that. This is a cross section of a bone. I mentioned that ring-like structure osteons, that you can see this link. This is an optical microscope image. And that small rectangle that I am showing in red is the area that we can go and do a scanning probe microscopic image. And we can do this in all the three directions. And that's what I'm going to show in the next slide. For example, you can take a section of the bone, cut it, and polish in three different directions. This is the transverse direction, and then this is the frontal plane, and this is the lateral plane. So you can make measurement on all three of this direction. Okay, like I mentioned, with the scanning probe microscope, on each side you can go and do an SPM image. This is, I probably think it's 5 micron in length scale, so maybe that just a minute. Yeah, this is the 5 micron length scale. So you can see this lamellar structure in the transverse plane. You can see that, and you can also, the indents being placed over here. You can see that small indents over there. So this is L1, L2, L3, three lamella that you can go and do the indents, both in transversal plane, as well as frontal plane, as well as lateral plane, and you can get the modulus out of it. Okay, here I'm given in the tabular column what the modulus are. In transversal plane, the modulus is much higher. It's about 22 GPa. In the frontal plane is about 15.6 GPa. In the lateral plane, it's about 12.5 GPa. And this is for 3 different lamellae. They are pretty much close, but you can pretty much see from this experiment that transversal plane has highest modulus, which means that that direction, it has the highest load carrying capacity elastically without undergoing a deformation, any permanent deformation. So this gives you an idea of how we can use these tools to understand the mechanical response of this material. Bone is a very complicated structure, and without this kind of imaging technique where we can go and image the surface and choose a site where you want to do this indents, it's very, very difficult, because in optical microscope, you won't even be able to see this structure and be able to measure those properties. That is the uniqueness of this technology, where you can go and do a scanning probe microscopy and get an image of the surface, and then you can select the site where you want to do the indent. That is the advantage of this technique. And you can do it at any orientation, and you don't need much of a preparation of the sample also. Here it's cut and slightly polished maybe, but you don't have to do anything else to do, because when you do the SPM image, you can go and choose the location where you want to do the smooth, wherever it is flat, you can go and select that location and do those experiments. Okay. So another thing is why this kind of a variation happens in bone material or bone-like material is mainly because of the difference in mineral content. Okay, so that's what I'm going to show in the next slide, where it's, instead of bone, we are going to use teeth material. Okay, that's for rat tooth. And what I mean by mineral is basically, for example, in tooth material, hydroxyapatite is the mineral, right? So depending upon the content of the hydroxyapatite, the mechanical property may be different at different sites. That's why you see the variation from site to site on this kind of biological biomaterials, hard materials. And it is same thing for bone. It is also same thing for tooth material, right? So in this experiment, this is the inside of the 980 Nanoindenter. You can see that transducers, there are two types of transducer. The big one is the high load transducer. On the left side, you can see the low load transducer with the scanner. This is the optical microscope and the Raman microscope attachment to it. So what I'm trying to show here is basically how you can combine different techniques to enhance your understanding. For example, if you want to understand what type of mineral is there, you need to understand the chemistry of it. So Raman is one way of understanding the chemistry that you can attach with this kind of instrument on top of what you're trying to do with respect to indentation or scanning probe microscopy. We can also additional instrumentation like Raman microscopy to understand the chemistry on the surface. So in this case, you can see the hydroxyapatite peak at 962 wavelength of 962, right? So if you can watch that, and then you can measure the intensity of that peak at different location, you know how much of that mineral is there on that particular site. So now you have the handle on the chemistry, you know what the content of that chemical is, or mineral is. So can we map that? If you see a higher intensity, which means the mineral content is higher, can you correlate that with the mechanical property that you measure? That is what we are going to see in the next slide, where you can see in tooth material, there are two things. That the one is the dentine and then enamel. Enamel is the hardest part and the dentine is the soft one. So when you chew something, you need to have both of this combination of soft and hard structure into it that is built into our tooth material, right? So dentine is generally soft. And if you look at the mineral content, basically that what I'm showing over here in this marking. The one is for what I think you are able to see my cursor. You can see that this is for the Raman intensity. It's an arbitrary unit. Higher the intensity means higher the mineral content. The other one that I am showing with my marker right now is the modulus of the material. Okay. So when you look at the dentine material, you can see the all red dots where the mineral content is high. The modulus is also high. The modulus is about 90 GPa. The modulus is very high. Whereas in dentin, the mineral content is also low, which is, you can see that intensity is low, and you can also see the modulus is also low. So now we are able to correlate the mineral content to the measured modulus. This is how we can go on progress in our findings. Okay, now we know there is a variation in mechanical property. Now we know why there is a variation, because the mineral content is. So we can tie the mineral content to the mechanical property. This is how we progress. Okay, like you mentioned this, yeah, the whole thing about this inundation on biomaterial is only maybe about 20 years at the max, or maybe 15 years. Before that, people use micro harness tester, but I mean, nano indentation itself is about maybe 30 years old. But using that for biomaterials is very recent. Like I mentioned in the beginning, even now, for understanding the soft materials, a lot of research is going on, even to build the tools as well as build the models to understand what we are making a measurement. So it is kind of still evolving, basically. Okay, so there is a lot of room to develop under biomaterials. Okay, so another example here is another tooth material. This is, in this case, is the strongest animals that we can find. For example, shark tooth material, and then we are also going to compare that with respect to piranha. And most of you know what piranha does is it has a very strong bite. You can chew anything. They are so strong. So we can go and understand what is the content of this tooth material, why they are so hard. Then we can understand kind of biomimetic, like can we replicate in whatever that we can find in the nature, artificially can we make it? That is the idea over here. So this is, I think this was done at Washington State University, one of our collaborators, where go and find this great white shark's tooth and do a cross section and polish it and do hundreds of indents. You can see that in this image. All the dots, what you see is like array of indents, and the color of those dots kind of gives you what the harness values of those the location. Because it's all automated, you don't have to do anything. Just by mapping where you want to do the indent and just say how many indents you want to do as an array, you can go and just press a button, the machine does it for you. So it's not a, it's a little bit time consuming, but it's all done automatically. So doing a property map is very, very simple now, and many cases it helps a lot for us. We get statistical average, and we also understand how the property varies on the topographic surface. Okay, so this is what's been done here. And on the left side, what I'm showing is the harness measurement and where the indents have been done. And you can also see the extrapolated map. Gives you an idea of what the harness on the surface is, how it is being distributed. Okay, it's very hard at the periphery, and as you go in interior, it becomes softer and softer. And this is on the right side. What I'm showing is the reduced modulus again as a map. Okay, what I'm showing over here is on the left side is the low displacement curve. If you look at the low displacement curve, and there are two different conditions. One is the hydrated condition, which means that you're doing everything inside a submerged solution. Normally we use saline solution like buffered phosphate solution, saline solution, and we do it in a hydrated condition. Okay, so if you do that for dentin, so for enamel material, you can see this curve here, the blue and the light blue. You don't see much of a difference in the low displacement curve for this particular specimen in the white shark. But if you look at the dentin material, you can see the red curve is in a dry state, and the pink curve is in a hydrated state, and it loses its property. Okay, you can see a big difference from dry state to hydrated state in the dentin material, but you don't see much on the enamel material. Okay, now if you do the same thing for piranha, you can also get the harness and modulus map on the cross section of the tooth material. And most of this experiment, the way we have done is basically the spacing between the indents are 50 to 100 micron apart, and the test the maximum load that been applied is about 1,000 micronewton, and they're all like load controlled feedback mode type inundation experiments. I mentioned that the experiments were done both in dry as well as in a submerged liquid condition. Okay, so now let's look at the average properties of those two type of specimens. One is the white shark, another is the piranha, and this is for the harness, and this is for the reduced modulus. On the left side is the harness, on the right side is the reduced modulus. If you compare that, you can see that on the enamel part, and both hydrate and dry condition, you don't see, there is a small difference, but not as much as you expect. Uh, and you can see both piranha as well as the shark has similar harness. You can see it almost, that's why they in the bite is pretty strong, we know that, right? And if you look at the dentin properties also pretty much comparable between piranha and white shark. And the same thing with the reduced modulus, you can see that. And then that when you do it in the dentin dry as well as dentin hydrated condition, you can see the difference between both. And then same thing with for the enamel as well. Okay, the modulus of piranha is almost compared to of shark. So I'm kind of giving an example like what all things you can do with this technology, right? Now you can compare like species of two different species, and you can measure the properties of this tooth material. You can measure the property of the dentin, you can property of the enamel, and you can do it in hydrated and dry conditions. And you can have some, you can develop some understanding. So all those things are possible with this kind of instrumentation. So far I've talked about hard materials, and now I wanted to change my gear and talk about soft materials. That is what I've been spending more time in recent times. And there's a lot of developmental work is still going on, understanding the soft materials, right? So one thing is when it comes for studying soft material, readily what people do is instead of using a sharp probe, they use a very flat-ended big probes. These probes are like a millimeter in size. This is a flat-end probe, basically a steel pin cut to polished to have a flat surface. So in those experiments, the vertical resolution can be still nanometer, but you lose the lateral resolution because now the surface area is much bigger. It's in millimeters, right? So we can still use it because if it is not very important that you need to do a nanometer lateral length scale, then you can still use this technique and get the low displacement curve and get an average property, okay? But we wanted to improve on further on that. That's what we've been doing for some time. One of the problem is as you reduce the smaller and smaller length scale, the size of the probe, then the contact mechanics and how you deconvolute the properties, it becomes a little bit complicated because of various other things comes into play. For example, adhesion. Okay, when you make the probe smaller and smaller, and the material is soft, you may not be able to even detect the surface of the sample. Those are all the things that I'm pointing it out in my next slide. So when you do measurement on soft materials, there are major challenges because the instrumentation that we have developed so far has been for hard materials, metals and ceramics. And when we pay attention, when we come to test soft materials, we need to pay attention to many other details. For example, how do you go and detect the surface of soft materials? For example, how do you detect the surface of water when you bring a probe to make contact? Because the surface stiffness is so low. And maybe measuring the stiffness, surface stiffness of the water is easier. Even some materials, you won't be able to even detect the surface of the contact. I mean, when you make probe and bring it to contact, you won't be even able to sense that. Your force resolution may not be there. So you have to redefine everything and then reinvent and develop instrumentation that is capable of having high force sensitivity. As the material are becoming very soft, when you apply very small forces, the displacement will be very large, right? So not only you have to have very high force sensitivity, you also have very high the range of displacement should be much higher than normally what you use for hard materials. So the probe should be able to measure large displacement jumps and large displacement excursions. So you have to have a high force sensitivity, large displacement range. So which means a different type of transducer need to design. And then the models that we have developed so far to understand hard materials, they are not going to work because they all assume elasticity, and when you do experiments on hydrogels and biological materials, it's all very complicated. They are viscoelastic, they are poroelastic. So the matrix and the pores, the combination of that, the mechanical response that you get is not elastic anymore. So you can't use those models that were being developed for hard ceramic and metals. So all these things you need to think about. So that's what we've been doing for the last 10, 15 years, slowly developing. So that's what I'm going to present in my slides, in the next few slides, right? So sometimes, like, I wanted to avoid mathematics and models and things like that, but when it comes to soft materials, you cannot avoid that. It all comes in hand in hand. It's not just measuring the low displacement curve anymore. You can measure that through instrumentation, but interpretation of those low displacement curve means that you need to understand what kind of interaction happens and how we can deconvolute the essential properties that we wanted to measure by using mathematical modeling. So it comes in. It is a dry subject, but we have to live with it. Okay. Now, so I did mention, I also presented up till now on hard material, the typical load displacement curves for hard material looks like that. You have a loading curve and unloading curve. From the unloading slope, you measure the stiffness. That's pretty standard, right? If you do this experiment on a hydrogel, okay, I'm hiding a lot of the various part of the curve. If you look at the whole curve, actually the whole curve just looks like this. And people who are familiar with the airform, they can readily see where I'm going with this, right? So let's say when you have a probe in air, it's not in contact with the sample surface, and you are approaching the probe towards the sample surface. At some point, you may contact, and normally you will jump to contact, okay? Because there is a sudden, because of the adhesion or and because of the surface energy, the probe will be pulled into the surface, or sometimes even the surface is so soft, they can bulge and make contact with the sample and with the tip. Any of those things can happen, okay? So when that happens, you make the contact between the probe and the sample, and then the force increases. Then you start to see the repulsive interaction. Before that, all that you are seeing is basically attractive interaction. So the probe is in contact. If you can follow my cursor, then jump to contact. These are all happening in adhesive interaction regime. And then as you increase the force becomes negative to positive, zero, and then positive. The positive force regime is what we call repulsive interaction, right? So normally the repulsive interaction is what we measure as the load displacement response and measure the properties of the material. But on soft materials, you need to measure the whole thing, okay? That is first mistake that people do. And when you do, and with the standard commercial nanoindenters, you buy them, they spit out this low displacement curve, and then you jump out and use the standard protocol and measure the properties. You're totally wrong. Okay, and that's what I wanted to point out. You have to measure the whole curve, okay, including the adhesive and the repulsive interaction. The second part is, like, if you look at this curve, that's what I'm clearly showing over here, and when the probe jumps to make contact, or the material jumps to make contact with the probe, this is at this point it happens. And then normally what we say, this is at the 0 point where we start to see the repulsive interaction, the positive force, and we define that as the 0 point. And the probe has already penetrated into the sample almost like 2 point, in this particular case, almost 2,800 nanometers, 2.8 microns. Okay, so this much of error in depth measurement is there in your measurement. So whatever the property that you are going to measure may not be accurate. Okay, another thing is, like, when you have this kind of material with high surface energy, you will see that in your adhesion measurement, basically there will be a large pull-off force. You can see that I mentioned that pull-off force over here. This is about 13 to 14 micronewton. And the maximum force that you applied in this particular experiment is also around 12 micronewton. So when you have a attractive force, adhesive force is about 10 to 12 micronewton, we cannot simply ignore that. Normally, when you do testing on materials like metals and ceramics, there will be a kind of adhesive forces between the tip and the sample because of the surface energy, but those forces are very small compared to the force that you apply onto the sample. So you can ignore that. Normally, that's what people do. But on soft materials, you cannot ignore that because it has a very large surface energy, and adhesive forces that you measure are almost on par with the forces that you may be applying. So you cannot ignore that. So the modeling aspect is totally different. You need to take care of all of these things. So I'm not going to bore you with all that modeling aspect, but I'm just giving you an idea that the methods that you have developed for hard materials may not work. That's why I divided my talk into two. When it comes to soft materials, you need to have a unique instrumentation and need to have a unique modeling approach to understand and solve these problems. Okay. So I'm going to skip some of these slides because this is not important. The mathematics is not important, but I just wanted to give this slide. This is not something new. This has been done in 1971 in Cambridge and Cavendish Lab by K.L. Johnson, where they simply used a very simple setup where you take a probe, it's a big probe in millimeters and millimeters in size, and then you have a transparent flat substrate, and then you put a camera underneath with an optical microscope so that you can see the probe from underneath, because this is a transparent substrate. You can see underneath as you make contact, okay, and then you slowly pull it out. Like you can measure the force through some kind of strain gauge load cell, and you can see what the force that you, as you pull this probe out, you can measure what the force is. At the same time, you can see all that contact in the optical microscope, and you can record how the contact is changing as you pull off. Okay, by measuring the change in contact size as well as the force when you pull off, you can get a curve like this. That is what I'm showing over here. Using this curve, they modeled that whole contact mechanics. It's called JKR mechanics. Okay, and surprisingly, this formulation works even at nanometer length scale. Okay, so that is the good thing. So we can use those kind of models, but we need to modify it a little bit. When we tried to do this in the beginning, we found some. I found some. This is part of my PhD, my postdoctoral work. We found problems. So we went back to Ken Johnson, who developed these theories. Then he tweaked his theory to match with what we found. And we were able to modify it that works even at nanometer length scale. Okay, so we have those models already there, which takes care of the adhesion in contact. So once you have that adhesion in contact has been taken care by model, then you can use this low displacement curve and then extract the mechanical properties. Okay, so not to bore you, I'm going to go skip some of these slides, and I'll go to the one important slide that I think I have here. Okay, so now there are three different methods that we can use. One is the way that I mentioned, that you have optical method where you can see the size of the contact. In my next slide, I will show you in some videos that you will get a clear understanding what I mean. Then you can measure the size of the contact through an optical microscope. And then when you have in this formulation, once you know what the size of the contact and the force you apply, and then use those formulation to calculate the modulus of the material. Okay, that is a pure optical technique. The other way is to measure the depth of indent from the low displacement curve, that the way I've shown you before, and convert this contact size into depth, and then use this formulation to measure the properties properly. The third method is by measuring the contact stiffness. I'm going to go and present you one more way of measuring mechanical properties, that is by force modulation. That was a part of my PhD work which I developed. And you can use also those approaches. So you have three different ways of measuring mechanical property. Essentially, the modulus of the material or the surface energy of the material using three different methods for soft materials. Okay, so now because we had a good understanding on this model, how we can use this model, we went and developed an instrument, and that's what I'm going to present. That is very useful for understanding the mechanical properties of soft materials, biological materials. So we called it as BioSoft In-Situ Nanoindenter. Okay. So this is the instrument, and this instrument basically is developed around a biological microscope, because most of the people who are going to use this type of instrument is going to be biologists, and it has been developed mainly to understand the mechanical response of soft materials, tissues, and cells, and hydrogels, and things like that. And you can use this optical microscope and all the optical microscopic capability, like for example phase contrast imaging or fluorescent imaging. You can use all of that to see the cells or the sites that you wanted to understand. And then you can see here there is a transducer hanging down on the condenser part. That is the part that we developed. This is a unique transducer which has, I'm going to tell you about more on this. And you can put that on a biological microscope, and then you can use the whole instrument now, not only see what you wanted to see in a biological system, and you can also do this experiments that we were able to do on a hard materials very precisely. That is what this instrumentation is about. Okay, so I mentioned that we need to have a unique transducer. That's what I am showing you in a schematic way. On the BioSoft transducer, it works in a different principle. Okay, up till now, whatever that I presented, we used electrostatic force actuator transducer, where you generate the force electrostatically and measure the displacement capacitively. That transducer is good, but when you try to understand the properties of soft materials, I mentioned that you need to have very large throw, which means very large displacement in hundreds of microns. That is not possible with that transducer, because the plate gaps, the spacing between those plates are only 60 microns or less than that. So you can't go beyond that. Okay? Only 1/3 of that gap is useful when you make a measurement. Okay, so we can't use the electrostatic force transducer for soft materials. We can do that if you are interested only in 5 micron or 10 micron depth range. If you wanted to do 100 micron range, you cannot use that. That's the only difference. So what we did was we built a transducer, and the way we did was basically it's kind of a folded structure, where you can have an actuator which can actuate up to like 200 microns, and that pushes a flexure, and the whole thing is measured. Basically, how much it is pushing is measured through a capacitance sensor. That's basically a 3-plate capacitance sensor with a bigger gap, so it can go up to hundreds of microns. So you can do that, right? And you can actuate and sense how much you are moving. Now, on top of it, you mount a load cell, and this is a very sensitive load cell made of capacitance transducer again. So that load cell has a spring, and depending on the load range that you want, the spring constant can be changed, and the deflection of the spring is measured using this capacitance sensor. So there are two capacitance sensors, and because the actuator can move 200 microns, this whole load cell can move 200 microns. Okay, now by changing the spring constant on this transducer, you can tune the load range that you want. If you are trying to do only on soft material, you can have a weaker spring, so that you have a very high force sensitivity. At the same time, you have very large displacement because you can move 200 microns. Okay, but if you are going to indent on a hard material, you can have a stiffer spring. You may have lesser sensitivity, but it can go for higher forces. So this is kind of a modular design. In all these cases, you can go up to a 200 micron throw. So that is the beauty of this transducer. The second part is The transducer that over here, which measures the displacement, is nothing but our electrostatic our transducer itself, which means that it can actuate and generate its force. So we call it as a null sensor mode, where we can try to actuate and keep the center plate at the center position all the time. At the same time, using this actuator, we can push onto the material. So now we don't lose the sensitivity. We have a very high sensitive force measurement. At the same time, you have a very large displacement range. Using this trick, we were able to build this transducer, and this transducer can be put onto any optical microscope. So this is how we were able to extend the range, both in terms of displacement, but not losing the sensitivity, force sensitivity of the transducer. This is what we call BioSoft Transducer. And then now this we can put it on, I mentioned that you can put it on an optical microscope. And on top of it, you can also change the type of probe that you want to put, because this is just a screw mount. You can unscrew it and put a different tip. So you can have a different type of probe. You can have a sharp probe, let's say tens of nanometers in diameter, or a very large probe in millimeter in size. So that's possible. Okay. And this is an inverted optics, so you haven't lost anything about your optical capability. So you can still see what is, if you have a transparent material, you can see when you make contact. So you can still use the optical capability. Okay. Now, I just want to skip that. So let me show you a video of what I mean, that you can see the size of the contact through the optics. So this is done in liquid. As the probe comes in contact, you can see that the liquid forms on the probe, and this is an inverted image, and you are seeing through a transparent surface. Okay, what you see over here is the low displacement curve, and you can see that it's a small jump in contact, load and unload. When you unload, you can see a large pull off, mainly because of the adhesion. Okay, so this both video as well as this low displacement curve is totally synchronized. So if you do 30 frames per second, you've got 30 points per second, and frame by frame you can take the video and you can analyze along with the low displacement curve that you make a measurement. So by this way, you can also have an optical measurement where you can measure the size of the contact. This is done so fast in this video, you are not able to see how the size of the contact is changing. But normally when you do it and replay it in a very slow speed, you can see how the size of the contact is changing. So that itself is in a measurement where you are measuring the size of the contact directly. You also have the low displacement measurement where you can use the modeling to extract the numbers. So in this one simple experiment, you have the way of measuring the size of the contact independently, but also you have a way of measuring the size of the contact through the low displacement curve. Okay, that is the advantage. Okay, now you can, because you have the optics, you can bring the cells in your view. This is osteoblast cells, and then you can bring it in contact with the sample that you wanted to study. So there is a way, if you have a fluorescence unit, you can also put biomarkers, and then you can use that to identify the cells, and you can bring into come along with the indenter axis. You can do a compression test. I'm going to show you one of those in the next few slides. So how you are making contact, how you are detecting it, there are two ways of doing it. In a quasi-static mode, what we do is, as you bring the probe, it comes in contact with the water or the liquid that you have in a submerged condition, and then we can measure the change in buoyancy, and then from the change of the slope, we can see where actually it is making contact. That's one way of doing it. I'm also going to present you a force modulation technique where we can use the phase shift as you approach as a way of finding the surface of the sample. There are two approaches. I'm going to show that in the following slides. And this whole thing, the BioSoft comes with all the models that I didn't present, but I mentioned. It comes with all the models. You can also plug in your own model. So along with the load displacement curve, you also have those as a package where you can go and use that and get the properties. Okay, so I'm going to go and give you some interesting findings. This is important. I think most of you probably understand this. One of the problems that we have in heart is the heart valve, right? And we all know that. And Sri Chitra is very famous with the Chitra valve. And basically, it's the aortic valve. Sometimes, due to birth defects, instead of having these three cusps valve, it may just have two cusps, or due to aging or some other medical condition, it may deform. For example, the function of this valve is to allow the blood to flow in only one direction. So it closes and it opens. That's what happens in our heart. And when it closes, it has to seal the whole thing, and the blood should not leak. And so it should be a very good close. When it opens, you should be opening fully so that the blood flows, so that the heart doesn't have to do extra work. So whether, depending upon whether it closes nicely without leaking or opens, the two conditions can give you rise to heart disease. Okay, and you have to end up surgically remove it. And when you do that, you have two choices. Either you can use a mechanical valve, or you can use tissue-based, animal tissue-based, or human tissue-based tissue-based valve. Okay. When you use a mechanical valve, the issue is it has a very high life cycle. It will go on forever, but the problem is the blood sticks to the surface, and you need to take the pills for blood thinning pills for your own life. Basically, all life you have to do that. And when you use tissue-based valve, you don't have that problem, but they don't have a long cycle of life, because then they eventually they wear out, typically about 10 to 15 years, and then you probably have to change the valve again. Okay, but you don't have to take that blood thinning capsules. That problem is not there. So one of our collaborator was interested in understanding, because nobody understood how the mechanical property is supposed to be when you have this the tissue-based valve. And it's very important, because this valve is closing and opening, and it's a mechanical action, and it's important to understand the properties, because if you wanted to build an animal tissue-based valve, you should know what should be the strength at different region. So that's very important important thing that we need to know. So it was surprising at that time nobody did this. So we were able to work with him to do that. And this is what I'm showing in this particular slide. And in this wall, there are three different regions. This is the region which is this is the edge of the region which is attaches to the cell. Okay, that is the blue one. And this is the mid region where you will have most pressure. It has to kind of withstand the pressure of the blood pumping. And this is the end region where it is free edge. It has to be very elastic, because when you're closing, it should conform to the surface so the blood doesn't leak. So which means that at three different regions, you need to have a different property. You have a gradient in properties. That's what you see on this low displacement curve. You can readily see that at the edge, for a given interpenetration, let's say 10 micron, you can see you require more load to penetrate 10 micron or higher depths. So which means that the edges, they are stiffer compared to the middle and the end. This is how it's supposed to be. Okay, and at the middle you can see it's less stiffer than the edge, and at the end it is very soft compared to the rest of the region. Okay, now we were able to kind of see that. Let me see that. So we can do a number of tests. It's very repeatable, and then you can see the properties in terms of modulus. Okay, so if you look at one of those, as I mentioned, from this low displacement curve, you can calculate the modulus. It is about 57 kPa, and that is for the edge region, which is the blue curve. And at the central region, it is about 37 kPa, and at the edge, in the free edge, it is about 10 kPa. So it is how it's supposed to be. So that kind of gives you an idea when you develop this kind of valve, what to expect at various regions and how to tune this property and to have the right function, how it's supposed to behave. So you can use this kind of instrumentation technique to understand that and then tune it. So this is, I thought it's a very important something that we were able to contribute to the biomaterial community. Now they have a tool to go and measure this and then fine tune their development. Okay. So now I'm going to jump into contact lens, and I think this may be slightly misplaced. Yeah, this is the contact lens. Okay, contact lens is also very important from the point of view. This is made of a hydrogel, and you are putting inside your eye, which means that it has to have a certain mechanical property. So when the FDA approved in US, they did approve with one particular test that the manufacturer have to do. That is, the modulus of the contact lens should be measured and should be within certain range, because when the modulus changes either low or high, when you wear it on your eye, it kind of irritates the eye, and it may not be a good thing for your eye. So FDA has put a specific number that when you make these contact lenses, it has to be within this modulus range in MPA. Okay. So normally in this lens manufacturer, the way they used to measure was to make a tensile specimen, like cut this specimen and put it on a tensile testing instrument and then measure the modulus. That's how they did it. But the problem is when you cut the specimen, you are destroying the whole specimen. There is no way that you can go back, right? So even if you store it in a hydrated condition over a period of time, that contact lens is gone. You already cut it, so you don't have it. So when we dealt with Johnson Johnson, who was interested in developing a technique to measure the properties of contact lens, we collaborated with them, and we were able to work with them to develop a technique where you can measure the properties of this hydrogel using nanoindation, depth sensing inundation technique. So we developed some kind of fixtures and jigs so we can put this lens in a conformed way inside a solution and bring a probe and make contact and make a measurement. Okay. So I probably be showing some of this commercially available contact lens and how we measure the properties using this BioSoft instrument. And it's very important to understand this part. Basically, hydrogels are mostly water, basically, okay? And it has a content of water as well as a network of structure. So when you do a stress, when you apply a stress, this water will squeeze out and come back. So a measured property that what you're measuring is basically a combination of fluid flow in and out as well as the matrix that it supports. So it's a combination of that. So when you make a measurement, you need to keep in mind it's not a straight elasticity problem, it's a poroelastic problem, and also has some viscoelastic component because the matrix is something which is viscoelastic material. So it's a combination of both, okay? And now, so using the BioSoft instrument that I presented, we were able to do the indents. You can see the repeatability, how repeatable it is. These are all like different indents done at different regions, but at progressively increasing load. And you can see the load displacement curves. And from the load displacement curve, this is HEMA contact lens. It's acrylate based. And there are two types of hydrogels we tested. One is silicon based, which I marked as SiHy, and the other is HEMA, which is acrylate based. And we were able to do the load displacement curve. And from the load displacement curve, and we can measure the modulus. The one thing that I want to point out is all these experiments were done in a hydrated condition, which means it's in a submerged in a buffer solution. So when you do that, you don't have much of an adhesion problem. This is the one trick people use. When you have probe making contact on soft materials like this, and dry condition, when you do, you have this problem of adhesion between the chip and the sample. Okay, to avoid that, they put it on, and most of the time you have to understand in a hydrated condition anyway. So, but when you do it in a submerged condition, you can minimize this adhesion. That's why you can see this low displacement curve have no attraction jumping in and jumping out, or any of those characteristics. I'll show that type of curve in real soon. Okay, so you can readily use Adhesion contact mechanics here and measure the modulus as a function of depth. Same thing here now with the different loading rate. Silicon-based hydrogel in this particular experiment shows loading rate dependency, whereas HEMA doesn't show much of loading rate dependency. Depends upon the formulation of how the contact lens were made. Okay, now we can, from I mentioned that you can use straight away Adhesion contact, slightly modified Adhesion contact, and measure the modulus, and you can get a map of it. That's what I'm showing over here between these two different materials. Okay, and you can also do another type of test on this hydrogel contact lens, which is a stress relaxation experiment, where you load the sample. Okay, that's what I'm showing in the loading curve, and then keep the displacement constant. Okay, so which means that the feedback is fighting to keep the displacement constant, and you measure how the load is relaxed. Okay, and you can measure the load relaxation as a function of time. Now you can take this load relaxation as a function of time curve, and you can fit it to a model. This was developed by Michel Owen, and you can extract the modulus of the material from this relaxation curve. There is another way of measuring the modulus of the material, and you can do it for different formulation. From that, you can get the modulus of the material. This is one way of doing it. That's what I'm showing over here. Now, you can also do it in a, this is also we did for HEMA and silicon-based hydrogel. I'm showing just some more information on that. Different type of hydrogel, but you can do the same studies. Okay. Now, when you do it in a partially hydrated state, like I mentioned, you don't get this nice low displacement curve the way that you wanted to get, right? When you do a partially hydrated or a dry condition, you will see this kind of low displacement curve. This is the curve that I started in the beginning. When you have soft materials, and you have this kind of curve where the tip jumps into contact and then load and unload, you cannot use those Adhesion contact mechanics anymore. You need to have this modified JKR method that I mentioned, and you can fit the unloading curve and extract the modulus of the material as well as the surface energy of the sample. You can do both of that. When we did that on this contact lens, and when we compared it with the modulus measured by Johnson & Johnson using their tensile testing, you can see the agreement between these two. This is the exactly condition that they do for the FDA approval, and one of those things that stopped them, because FDA kind of mentions this is how you have to measure the modulus. When they tried to measure it with the indentation-based technique, it didn't match with what their number they were getting with the tensile testing. But when we did with this method, with modeling approach, when we compared the modulus, it matched pretty much close to what we expected with the tensile testing measure. Now, so they are trying to figure this out with the FDA to get it approved for this kind of material testing technique. See, so far
pardon me, Dr. Syed.
Yep. Yes. I can hear you.
Pardon
Go ahead.
me, Dr. Syed.
Yes.
This is Dr.
Yeah, Ajit, we can hear you.
We can hear you.
Another 5 minutes for the session, after which there will be many questions from the participants.
Yeah, sure. I'm going to almost finishing it now. Okay.
Yes.
Okay, very good.
So, yes, sir.
Okay. So in this, one thing that I wanted to extend it to stiffness-based measurement, I mentioned that you can also apply, the method that so far I mentioned is about quasi-static, which is basically you applying a continue force in a quasi-static manner and measuring the displacement in quasi-static manner. In dynamic method, what you do is basically you apply a sinusoidal force and measure the sinusoidal displacement. And from the force that you apply and the displacement you measure, basically the phase shift between these two, you can see whether the material behaving in a viscoelastic manner or not. For example, when you do this on an elastic material, the force you apply and the displacement you measure, there won't be any phase shift between these two. The phase shift will be zero. But viscoelastic material tends to have a phase shift. That the degree of phase shift gives you the idea of the viscosity of the material. This is something that I developed during my postdoc work at Naval Research Lab that will later be commercialized at Hydron. And using this approach, what you can do is you can modulate the tip, which means that you are applying a sinusoidal force to the tip and bring the tip close to the surface and try to measure is there any phase shift between the tip and the sample, okay? If you see a phase shift, it's mainly due to some kind of attractive forces, okay? For example, when you run this tip at the closer to resonant frequency and bring the tip closer to the surface, any interaction will shift this phase to either right or the left side, depending upon whether you have attractive interaction or a repulsive interaction. And this is a very sensitive measurement technique, okay? By monitoring the phase shift, you should be able to say whether the tip is making a contact or it is away from the surface. Even when the surface forces are acting, even before the material is in contact with the specimen, you should be able to feel the tip by measuring the phase shift. That's all I wanted to mention about this slide. So measuring the phase shift is a very sensitive way of sensing the surface, and using that, you can do a lot of good things. One thing that I would like to show you is how you can do a compression test of a living cell, okay? Using a fluorescence microscope, which comes with the instrument when you buy a fluorescence microscope option, you can put a biomarker, you can see the cell, and if you use a very flat ended tip, like in this case is about 100 micron, you can bring the tip, you can and then pretty much bring the tip in alignment with the cell that you wanted to do the test, and then make contact, okay? Now, as I mentioned, making the contact and sensing the contact of the cell is a very challenging thing. When you make contact, you won't feel like you are sensing the contact. So that's why you need to use this phase approach. So if you monitor the phase, for example, in this next slide, this is done in a submerged condition using a PBS solution. And in this, you see the whole probe is being submerged, and you can see that cell is just beneath the probe, and you are approaching with the probe to make contact. So if you look at here, the phase shift as a function of approach, when there is no cells, like when you make directly contact with the glass slide that you have, the phase shift shoots up, okay, from minus 160 to 0 degree. But when there is a presence of cell, it phases down because of that attractive forces. The phase shift is moving in another direction, and you can see a big jump. Now we know that the tip is getting attracted to this cell structure that we can see through the phase shift. So we know that we are making a contact, and then it kind of makes contact, compresses the cell, and then you can see that it comes back. This is during this compression, there is a small change in phase shift, but then it jumps back as soon as the tip makes contact with the glass slide again, which is similar to this with the red curve that you see. So using this approach, we know when we are making contact with a cell, and then we can also extract the forces that we measure. And in this low displacement curve, you can see at some force, like 4 micronewton, the cell is busting. So this is done on a live cell. We were able to measure the force that is required to bust the cell, okay? Something the biologists wanted to know. By measuring the phase shift, we were able to do this approach and make contact, and we were able to measure that. So that's why I wanted to put it over here, okay? Using the same approach, we can also measure the mechanical properties. For example, not only measuring the low displacement curve, but also measuring the stiffness as the tip approach make contact. You can measure the stiffness as a function of approach distance. And if you plot that, you get this curve, which is basically interaction stiffness as a function of interaction force. by the model that I presented earlier on, you can fit the model to this curve and you can accurately measure the modulus of the material. This is another way of measuring the modulus of the material for soft materials. Here, you don't have to worry about where the tip is making contact because everything is captured in this stiffness measurement technique. So you can just have to just fit the curve and get the modulus of the material. So the worries about where we are making contact, where is our 0 point, those things you don't need to worry about. Okay. So using this technique, we were able to measure and compare it with other technique that works very well. So with that, I'm going to stop here because I think it's time. I'll take any questions. So with the BioSoft material, we were able to extend our measurement technique to very soft materials, and you can go and do a very large displacement range. The force sensitivity is good enough to do this measurement. And it comes with the data analysis package that you can directly use and measure properties. I have a couple of slides. There's some last slide that I would like to show. There is, I mentioned in the beginning that there are other ways of measuring mechanical properties of soft materials. AFM is one of them. In this particular setup, in this Bruker AFM, it has the capability of not only measuring the forces and displacement using the AFM, it also has an optical tweezer with which you can trap the cells and do cell manipulation. At the same time, you can do AFM measurement. I thought something unique. So I will stop with this and then take any questions. Okay, so over to you.
Thank you, Dr. Sayed, for the very informative speech.
I'll
So
just
I'll be posting
okay,
the questions on behalf. Yes.
Okay, let's stop the presentation. How do I do that? Or you
So
can
Ajit can read out the questions to you and you can answer.
Yes. I'll read out the questions. Yes.
okay.
So Ritvik has asked, can you suggest some instruments to measure the stress induced on thin films of biomaterials deposited on some metal substrates?
What is the stress induced? What is the
He
induced
wants
stress?
to
Can you measure the induced
stress,
stress on a thin film?
yes, on thin
Well,
film of biomaterials which are
okay.
deposited on metal substrates.
Yeah, you can sense if there is any residual stress that is being introduced when you do the deposition, you can sense that through the low displacement curve. But if you ask me whether you can measure that stress, it's not that easy. Okay, a lot of work has been done in this field to measure the residual stresses, uh, but it happens basically the contact geometry itself changes that the harness doesn't change with the residual stress. But the low displacement curve will give you an indication whether you have a tensile stress or a compressive stress. When you have a material that doesn't have any stress, when you compare that low displacement curve with the curve that you have, you think there is a residual stress, there will be a difference in the low displacement curve. Okay?
Thank
You
you.
can use that to identify.
Yeah,
Another question from Dr. Father Arun Jose. He is asking whether the Nanoindenter has a real advantage over micro-hardening test used to analyze mineral composition range.
Definitely. With the micro indentation technique, which uses optical technique, you only measure the hardness of the material, and it is so it is limited with the resolution of the optical microscope. Whereas in depth sensing nanoindentation, you not only measure the hardness, you can also measure the modulus. There is a lot of other things that you can measure. I didn't mention anything in this talk. Basically, you can not only measure hardness and modulus, you can also measure tribological properties. You can measure fracture toughness. You can measure tree properties. All those things I didn't cover in this talk, but all those things can be measured using depth sensing nanoindentation technique. Fatigue, all of that, which is not possible with micro hardness tester. Okay.
So yeah, additional part to this question also is there, Ajit. So he's asking basically, is this instrument for heart tissues invasive or non-invasive in use?
This is basically size scale dependent. When you say nanometer, tens of nanometer, is it invasive technique? Depending upon what you're trying to measure. If you have like a 1 nanometer thin something, and if you're doing a 10 nanometer, it's an invasive technique. But you have like tens of microns and you do a couple of nanometers, I would say it is non-destructive.
Okay. Another question from Mr. Harikrishnan.
Mm-hmm.
So can we obtain elasticity metric?
Yes. So, for example, if you wanted to understand the anisotropic anisotropy on a single crystal or a surface where or a plane where you wanted to measure, you can do that. So, for example, when you use different type of probes, like a new probe that has elongated axis, you can use that to sense the anisotropic. You can do that.
Thank you, sir. Another question from Harikrishnan himself.
Mm-hmm.
So can we use the same indenter instrument for hard biological materials and engineering materials like metals and ceramics?
Yes, that's not an issue at all. Hard materials, hard biomaterials, metals and ceramics, you can do that with our standard instruments.
Sir, can I add one question?
Mm-hmm.
Sir, I'm Harikrishnan. My question is, can we find elasticity metrics with a single indentation? That's my question. Is it possible for powder or bulk materials?
With a single indent?
Yes, sir. Yes, sir.
With the single indent, if it is depth dependent, you can find that. One single indent in one single location, as you indent, you penetrate at different depth. If you see a gradients in mechanical response, you can sense that.
Okay, sir. Okay.
Okay.
Okay. Okay.
If anybody wants to pose questions one-on-one, you can do that.
Hello.
Yep.
Asif, this is Kiran.
Hi, Kiran.
Hi. So it was very interesting and a lot of information is here. New information I have seen in the presentation. Great. How do you calibrate the machine compliance? Actually, I want to know how do we manage when particularly we want to do with the soft materials like gels and all. So compliance plays a major role, right?
Well, for soft material, it's not of a big issue. And usually we measure the same method that we normally measure machine compliance. And because the issue with compliance is big when you have materials with a high modulus, right? So when you have a high modulus, you are also deforming the system, right? So, but when you have a soft sample, when you indent, your deformation of the machine is not as much as compared to when you indent with a hard sample. So compliance is not a big issue, but still we can measure the compliance of the instrument by doing the standard procedure that we normally use. It does, for example, when you do it on a BioSoft, you can map the whole stage area where the compliance is changing, which we have already done, and it's not a big deal at all. It's not an issue at all. Compliance.
Right.
Yep.
Sure. Thank you.
Are there any further questions?
Okay.
I hope not. Yes. Yes. Are there any further questions?
So I think
Okay.
let me share my last slide, which has anybody wants to know more about it, they can always contact me on my email address or Pratyang. I've given both of our email address, so you can reach out to us. And okay.
Yeah. Also, we have a R&D section.
Yeah. So I forgot to mention, we have an R&D center in Trivandrum Technopark, where we have all this instrumentation that I mentioned, except for the BioSoft. We have the standard instrument. And the way we do is basically we are very good in collaborating or interacting with people. Like we have been ongoing thing that we do with the Indian Institute of Science or ID Mumbai. We like to interact with people and do, particularly on these biomaterials. We are very interested. We are doing something now with Manipal University, so we are very open to that. So you can always come and visit us at Technopark. Not now, maybe later after COVID is done. So we are there here in Toronto. Okay.
Thank you, Dr. Sayed. It was really informative, and it was a really honor to host you for this particular session. And it was
My
a lot of knowledge shared in a short frame of time.
my pleasure.
And thanks for your time that you have spent from a completely different time zone. Thank you very much.
Thanks for
Thank you very much.
patience to sit down and listen to whatever that I said.
We were thinking maybe a little longer, but yeah, 1.5 hours.
I
It
didn't
didn't really realize.
I didn't realize at all.
No, it was fine. It was fine. We concluded right in time.
Okay, that's good.
And for the participants, Dr. Syed is truly an alumni of distinction for his alma mater, and he is truly a citizen of distinction for our nation. So I really feel privileged and honored to have him in this session for today. Thank you very much, Dr. Syed.
Thank you. Big
Thank you.
words, but thank you.
Great.
Okay. Thanks you.
Okay.
Thanks for everyone to coming and attending for this conference. Okay. Stay
Yeah.
safe.
You too. You too.
Thank you. Thanks. Bye bye.
Bye.
Bye bye.
Bye, everyone. Thanks.
So for the participants' information, please don't miss the next session. Very important session for us. A very distinct or distinguished faculty, our Professor Bikramjit Basu from IAC Bangalore, recipient of highest teaching award, Shanti Swarup Bhatnagar Award, will be presenting his vast expertise in a short while. So please don't miss the session. Let's come back 11:45.